The term gymnospermae is built from two Greek roots, gymnos, meaning naked, and sperma, meaning seed. Advanced angiosperms encase their seeds inside an ovarian wall, which eventually ripens into a protective fruit. Gymnosperms leave their ovules completely exposed to the elements. This yields an absolute anatomical rule for the group. A gymnosperm ovule is never enclosed by an ovary wall before, during, or after fertilization. Earlier plant forms relied heavily on standing water to transport motile sperm during reproduction. To expand into dry terrestrial environments, plants required a biological workaround to bypass this aquatic limitation. Stripping away the ovary was a precise evolutionary adaptation. By exposing the seed and altering the mechanics of fertilization, these plants rapidly spread across the arid land masses of the Mesozoic era. The macroscopic, visible body of a gymnosperm is the diploid sporophyte. A sporophyte is the diploid, spore-producing phase of a plant's life cycle. The scale variance within this group is massive, ranging from dense knee-high shrubs to the tallest living organisms on Earth. The absolute physical extreme of this towering architecture is the giant redwood, Sequoia sempervirens, dominating the forest canopy. This massive scale represents a complete structural inversion from earlier terrestrial plants. In mosses and liverworts, the small haploid gametophyte was the dominant phase. In gymnosperms, the massive diploid sporophyte takes total control of the life cycle. Surviving on dry land meant enduring severe temperature fluctuations, high-velocity winds, and critically low atmospheric humidity. Gymnosperms counter these threats with highly specialized xerophytic foliage. Xerophytic refers to plant adaptations explicitly engineered to prevent water loss in arid environments. Many conifers grow leaves shaped strictly as needles. This tight, compact geometry drastically reduces the surface area available for evaporation. These needles are coated in a thick, waxy outer cuticle layer that physically seals moisture inside the plant tissue. They also utilize sunken stomata. Stomata are the microscopic pores used for gas exchange. By recessing these pores deep into the leaf structure, the plant traps a boundary layer of moist air, preventing high winds from stripping water away. Supporting stems follow specific architectural patterns. Species like Cycas maintain strictly unbranched stems, while conifers construct heavily branched networks to support their needle arrays. Combined, the reduced surface area, thick cuticle, and recessed pores form a physiological fortress against transpirational water loss. Below ground, the taproot system faces the distinct and constant challenge of extracting essential nutrients from barren, rocky soils. Species of Pinus solve this through mycorrhiza, a mutually beneficial symbiotic association between a plant root and a fungus. This web expands the root surface area, pulling in water and minerals the plant couldn't reach alone. Cycas takes a different approach, developing specialized coralloid roots. These nodular structures house internal pockets of cyanobacteria. Cyanobacteria are photosynthetic bacteria capable of fixing atmospheric nitrogen into a chemical form the plant can consume. By outsourcing nitrogen fixation and water absorption to specialized symbiotic partners, gymnosperms successfully establish themselves in nutrient-poor terrestrial soils. Reproduction in this group is governed by a strict biological rule, heterospory. This is the production of two distinctly different types and sizes of spores, small male microspores and large female megaspores. Both spore types develop inside protective chambers called sporangia. which sit on modified leaf-like appendages known as sporophyls. These sporophyls pack tightly together, spiraling along a central axis to construct compact strobili. We commonly refer to these structures as cones. A male cone is built from microsporophyls, which produce the male microspores. A female cone is built from megasporophyls, which shelter the female megaspores. In a pine tree, you will find both the male and female cones growing on the exact same plant. In a cycas, male and female cones are segregated onto entirely separate trees. Organizing distinct male and female spores into compact, specialized cones establishes the architectural prerequisite necessary to produce a true seed. This diagram illustrates the gymnosperm life cycle, highlighting where the diploid sporophyte generates the haploid gametophyte via meiosis. Following the male pathway below, microspores divide to become a highly reduced male gametophyte, a pollen grain. On the female pathway, a megaspore mother cell divides meiotically. Out of the 4 resulting cells, only one survives to mature into a multicellular female gametophyte, which bears the egg-producing archegonia. Unlike the free-living gametophytes of ferns and mosses, the gymnosperm gametophyte has 0 independent existence. It remains permanently trapped within the parent sporophyte's tissues. To cross the physical gap between trees, gymnosperms use anemophily. They rely entirely on wind currents to carry pollen grains to the female ovule. Once the pollen grain lands, it builds a pollen tube. This specific mechanism, known as siphonogamy, allows the tube to physically penetrate the archegonia and discharge the male gametes directly to the egg. The fusion of these gametes creates a diploid zygote that will grow into an embryo. The exposed ovule then hardens around it, completing its transformation into a naked seed. Note carefully, the nutrient-rich endosperm within this seed is haploid in origin. It develops from the female gametophyte before fertilization even occurs, a stark contrast to the triploid endosperm found in flowering plants. By internalizing the gametophyte and engineering the pollen tube to deliver sperm, gymnosperms successfully severed plant reproduction's ancient reliance on liquid water. Taxonomists divide the surviving gymnosperms into 4 distinct classes. Psychopsida includes primitive, palm-like plants like cycas, easily identified by their strict, unbranched stems. Coniferopsida is the largest and most dominant group. It contains the massive evergreen needle-bearing trees such as Pinus and Cedrus. Ginkgopsida contains exactly 1 surviving species, Ginkgo biloba. Known as a living fossil, it is immediately recognizable by its unique fan-shaped leaves. Finally, Netopsida includes species like Ephedra. This class is an advanced evolutionary outlier because it possesses xylem vessels. Xylem vessels are specialized water-conducting tubes that are otherwise strictly reserved for angiosperms. This specific stratification of classes represents a vast evolutionary testing ground, bridging the physiological gap between primitive spore-dispersing ferns and highly advanced flowering plants. The biological architecture of gymnosperms extends far beyond the forest ecosystem, driving massive sectors of human industry. Conifers act as the global primary source of softwood timber. Their lightweight, durable wood provides the raw material for commercial construction, furniture, and the bulk of the world's paper pulp. Their specialized biology also produces crucial chemical outputs, including commercial resins and turpentine extracted directly from species of pinus. They offer niche nutritional resources as well. We harvest their edible naked seeds to produce pine nuts, known as chilgoza, and extract rich starches to make cycas sago. Medically, gymnosperms are an invaluable resource. The chemical ephedrine is extracted from ephedra and utilized globally to treat severe respiratory issues like asthma. From the bark of the taxus tree, biochemists isolate taxol. Taxol is a potent chemical compound deployed in chemotherapy to actively freeze the division of cancer cells. The gymnosperm is a highly specialized survival machine. By engineering the naked seed and mastering the harsh terrestrial climate, these plants establish themselves as a foundational pillar of both the global ecosystem and modern human civilization.